This is a walkthrough of using Tablet. Tablet is a next generation sequence viewer produced by the James Hutton Institute. If you need any help when using Tablet, then there's a little red button up at the top right. If you click that, a web browser will open with the appropriate help pages and the manual. So we want to import an assembly into Tablet, and if you've worked with assemblies before, you'll know that they can be hundreds of megabytes in size, maybe even gigabytes, possibly even terabytes for large assemblies. You don't want to be dragging those out and installing them on your local disk, so Tablet will let you read them over the web from a single repository, so many of you can explore the same assembly at the same time without duplication of data. So we click the import assembly button. I'm going to paste in a URL. This is a local URL for Dundee University on our teaching server. It's not accessible off campus, so please don't try, you won't get to it. And I've pasted in the address for the BAM file. This is a sorted, indexed BAM file. You have to work with a sorted BAM file with the appropriate index so it can find the data and access it in a credible manner. You do need the .bai file in the same location as the original BAM file. And we load in the reference sequence k12.fasta. This particular assembly is an 0157 dataset assembled for teaching purposes against the K12 reference. So we can then click open, and Tablet is now opened. It says select a config to begin visualization. We have one config. You can find this in the panel on the left hand side. You can expand that a little, you can drag to be able to see all the names, you can see the length, number of reads and so on. All draggable, configurable. We've got the main canvas here, and if we select a config, it will then open up. We'll see the distance that we're viewing. In this overview window, we've got the region from 1 to 25,000 in our alignment. The actual alignment is about 4 million long, I think, the total length. You see this chromosome, 4,600,000 odd long. So Tablet just opens up a chunk at a time, and if you move on by chunk with this slider at the top, let's go somewhere near the middle, it'll go off over the web, collect that chunk out of the alignment file. Below that we've got the DNA consensus sequence and translation for the first frame. We'll configure that in just a moment. We've got a coverage map, and we've got cigar features illustrated in the sequence there. We'll load features in just a moment. So the main tools are all found up on this ribbon at the top. We've got a home where we can do various things. We'll come on to those in a moment. We've got different color schemes we can choose, and we've got an advanced tab for some of the more advanced things. We're not really looking at gene sequences. Let's turn off the protein frame. You can actually turn on whichever frames you want, so both forward and reverse. There we go. And now I'm just going to turn them all off again. There's the consensus. We can get various different views on this. We can zoom, scroll along, just drag the button on the bottom, and you can see a little window moving in here. You can see that there's quite a deep assembly, so we can scroll up and down through that. This is just using the mouse button, mouse scroll wheel, or dragging the slider on the side up and down. We can also just hold down Control, use the scroll wheel, and this will help us zoom in. Back to the home. Plenty of different things that you can do to customize this as well. If you want to zoom in more, you can use the zoom button on the slider on the top to zoom right the way out to your consensus read. You can drag the window that appears, the little box, to go to the most appropriate part. It can occasionally take a little while to catch up, upload the relevant data. And if you want to be able to see variance, points of change, you can just darken the plot and the variance will start. Let's zoom in just a little bit. The variance will start to stand out. Other things you can do, zoom in a bit more, interesting bit, let's just double click and it will zoom us in a little bit there. So we can do the variant highlighting with the variance. In fact, you can go to almost a completely sort of Manhattan at night view there, where just the key changes against the consensus are shown. We can also choose a tab and colour the various. Let's go back and make that a little bit lighter, shall we? It's a bit on the dark side. Let's go to about the middle. That's quite nice. Go back to the colour schemas. Colour by the direction of the read. Colour by the read type. So let's move over here. So we've got properly paired reads. Reads that aren't properly paired will be coloured differently. So we've got those forward reads that are properly paired are green, reverse reads that are properly paired are blue, and then other reads will be, I think, red and another colour. If you've got different read groups, you can colour by read group. Here it's not really colouring by any read group, it's just highlighting the variance, which you can also do with the variance selection there. So what the variance does, it takes the alignment. Where are we up here? We've got a window here that we can drag up, and you can see it colours every base grey that matches the consensus, and those that don't match the consensus are highlighted with a red colour. So looking at this, we can see putative features. Let's load up some features ourselves. So let's go to Home, click on Import Features, and we will, there we go, it's popping up there. We will go and find some that I prepared earlier. Give me just a moment to pull these ones up. Navigate through to find a variance VCF file. So this is a standard format for representing variance in an assembly when mapped against a reference. So we'll open those, and Tablet will then tell you how many references it's been able to upload. Unfortunately, the pop-ups don't appear on the video for some strange reason, but this has imported 59,806 variants. So where are those variants? Well, if we zoom out, we can see clearly where we would expect to see some, and we can select the visible tracks above by right-clicking and taking Select Visible Tracks. And then a pop-up will appear that unfortunately you can't see on the video, where you can select the appropriate variance types you want to see. I've deselected everything except for snips. So let's click that, and now we see we have a snip track. That looks like a snip. Let's scroll in a bit, and there we go. We can see these various snips, and they're highlighted by green triangles on that track. A couple of other things: read packing, and couple of options. Packing will try to pack them in as best it can. Stacked, it will just start to lay them all out in a what I think is a not terribly usable form here. I really don't see much of the point of using a stacked approach. I much prefer the packed, but that's me. Others will have a different view on that. So now I can scroll around, find the variants I want, see the tags, right click, and see what there is available. Just hovering over the green triangle will pop up a pop-up. You can't see it on the video. It's something to do with how it captures the information from the screen, but that tells you what the various parameters for that particular snip are. You can see the whole of the VCF entry for that particular line.